And when I say everyone, I say everyone. Alright, so in the previous class, we had discussed about the various steps that are there in the cloning, what cloning is and everything. So now, today we're going to go into depths of how the process of recombinant DNA technology, or you can say how the process of the whole entire genetic modification is done. So we have already discussed that the first recombinant DNA, it was made by two scientists together, Stanley Cohen and Herbert Boyer in the year 1972. Now what they did while doing so, they linked a gene encoding antibiotic resistance with a native plasmid of Salmonella typhi. This is what they did, and their experiment was actually very successful. So they received a Nobel Prize for the very purpose. So in the year 1972, so you people, this can be asked in the question as though, like, in which year was the first our recombinant DNA made? So you should know that it was made in the year 1972. Now, there are three basic steps in genetically modifying the organisms. Now, talking about what are those steps? See, the first thing that a person or anyone that is genetically modifying any organism, the first thing is that the person should identify the DNA, whichever DNA, sort of like the gene of interest, whatever is the DNA of interest or the sequence of interest. Now, who can tell me what is a genetically modified organism? Genetically modified organism, GMO. Who can tell me what is it? Genetically modified organism. Or genetically modified organism. The name, like the answer lies in the name only. Tell me.
The genes are modified.
The genes are modified. So meaning those are animals that have their genome or DNA sort of altered or modified, isn't it? Yes, exactly. Very good. So those organisms will be called as the genetically modified organisms. Now remember one thing, it can be a bacteria, it can be, you can say, an animal, it can also be a plant. So the GMO can be anything. Now, so while we are trying to genetically modify any organism, what we have got to do? We have got to, first of all, identify the DNA of interest. Like maybe, for example, if you want to make a new product from the old one. So the old one must have something that is in like of interest to you. So you want to extract that. And that is actually done when we identify what is the desirable gene. Then what we do is that we introduce that identified gene, meaning the gene that we have taken out. Now we are going to introduce that particular gene into the host's body. Lastly, now once we have done all of these things, now we want the host to make more and more particles. That is the whole, you can say, um, concept behind it. So now we have like basically maintenance of introduced DNA. Now the DNA has been introduced in the host and then it has to be transferred to the progeny. Because if the host will divide, so our desired DNA will also divide. And then it will go to the progeny. So we can see the generations and everything along with it. So maintenance, like when whatever DNA is getting inside the genetically modified organism. So if it will get lost after one generation, then it's of no use to us. The whole entire experiment is failed. But in this particular case, wherein we maintain the introduced DNA, if whatever DNA has been introduced, we will maintain it. Okay. Now, to do all of these processes, like these are the three, you can say, basic processes that we have got to do. So to do all of these different processes, there are certain tools that we require. So this is sort of like a brief of whatever tools we need during making of the recombinant DNA technology. Now, the first thing we need is the DNA manipulative enzymes. We need a variety of enzymes, basically like a set of enzymes we require for recombinant DNA technology. So the first type of DNA manipulative enzyme is the restriction enzyme. Very, very, very important. Okay, restriction enzymes are required. Then there is polymerase enzymes. How many of you remember PCR? PCR. None of you remember PCR? What is the full form of PCR? Yes, none of you. The full form of PCR? It stands for polymerase. Why is no one speaking anything today? No one is like bothered to speak anything. Why? You people are not in the mood to study? If you're not, tell me. I'll go. Are you in the mood to study or not?
Yes.
And the rest of you are not. No one is saying yes.
Yes, ma'am.
If you are not going to respond in the class, how will you learn? You're getting my point. Amna, you never speak in the class. The only thing I ask you people is yes or no. That also you don't speak. So then I don't feel like teaching. Then I feel like everyone is getting bored. Why should I teach? They're like, they'll not learn only. Please be responsive in the class, all of you. So, yeah, I was asking about PCR. You people don't remember PCR? I've taught you PCR, right? Polymerase chain reaction, wherein we used to amplify the DNA. Anyway, so polymerase enzymes are also required for the recombinant DNA technology. And then comes the ligase enzymes. So these are the set of enzymes that are required. Then the next requirement under the recombinant DNA technology is the need of the vectors. Vectors are the carriers. So we need vectors. And lastly, where are we going to put the gene of interest, put the vector into? So we need the host organism. So this is the necessity for the recombinant DNA technology. Now we are going to discuss the first type, or you can say the enzymes. The one that we are going to discuss right now is restriction enzymes. Now, if you say restriction enzymes, or in a more layman language, if you want to say molecular scissors, not the normal scissors, but the scissors that are, you can say, inside our body, right? Molecular scissors, or you can also call them as chemical knives. So these are the particular enzymes, particularly proteinaceous structures that cut the DNA into fragments, meaning a long strand of DNA is there. Whenever the restriction enzyme is applied, so it will cut the DNA and the DNA fragments will be present as such. So they cut the DNA into fragments. Now the restriction enzymes, they belong to a larger class of enzymes called as nucleases. So it belongs to nucleases. Now in general, the nucleases are of two types: exonucleases, endonucleases. Now by the meaning, or you can say by the name only, you people can make out exo means out, endo means inside. So whenever, as I told you, that what is the function of nuclease or restriction enzyme in general? It is to cut. It makes cuts. So if it is an exonuclease, okay, exo means towards the outer side normally. So now this exonuclease, what will it do? It will remove the nucleotides from the end of DNA molecule. So if this is the DNA molecule, so it will remove from here. It will not attack in the middle. It will act on the end. So that becomes an exonuclease. Then comes endonuclease. Endo means inside. Nuclease is it is an enzyme. So what does the endonuclease do? It breaks the internal phosphodiester bonds. It breaks the internal phosphodiester bond. So both of them belong to nucleases, but both of them have different functions. Okay, and tell me one thing, students, where is this phosphodiester bond present? Have you ever heard about this before? Phosphodiester bond. Where is it present? Have you ever heard about this or not? Yes or no? Tell me first that.
No, ma'am.
No, none of you have heard phosphodiester bond. Okay. So the two strands of DNA molecule, they have phosphodiester bond in between them. I taught this in molecular biology chapter to all of you. But since you people don't study, of course you'll not know. Okay, now talking about the first restriction endonuclease. See, obviously, since it is an enzyme, it is a chemical. So you can think about that. Okay, it may have been extracted from somewhere, right? It may have been taken out from somewhere. So same way, the first restriction endonuclease that was isolated had a name Hind 3. Hind, sorry, 2. Hind 2. It was the first endonuclease that was isolated. Now, what does it do and how does it actually cuts the DNA? So, Hind 2, what it does is that it cuts the DNA molecule at a particular site. I mean, if you take a DNA and you add Hind 2, it doesn't mean that it will cut the DNA from everywhere. No. Hind 2 usually cuts the DNA at a particular point. And how does it do that? So it cuts it at a particular point by recognizing a specific sequence of 6 base pairs. So Hind 2 goes to the area that has 6 base pairs sequence, or the specific sequence of that 6 base pair. And it goes and attaches over there and then it breaks the bond. So remember one thing, students, that there are so many different kinds of restriction enzymes. Some of them I have mentioned later on. So it's not necessary to remember all of them, but one or two you should remember. Now talking about the like how many restriction enzymes are there and from where we have got, like where did we get them from? So remember that there are over 900 restriction enzymes, over 900 restriction enzymes that have been isolated, and that too from over 230 strains of bacteria. So imagine E. coli is one, right? E. coli is one, Salmonella typhi is two, like that 230 bacteria strains. And from them, 900 restriction enzymes have been isolated from the bacteria. Now, this line tells us, okay, tells us about how many restriction enzymes are there and from where it has been isolated. So now we know that restriction enzymes, they are obtained only from prokaryotes. And under prokaryotes, which one? Bacteria. Now it is, now how do they have this particular enzyme and we don't? Because they have this natural defense system. They have to protect themselves from the action of bacteriophage. You people know what a bacteriophage is? Bacteriophage is a virus which infects bacteria. See, there are certain viruses that infect humans. There are certain viruses that infect the cattle and stuff. There are some that infect variety of people like that. Right? Not people actually, but the viruses. So same way, the bacteriophage. Bacteria refers to the bacteria. Phage means eating. So bacteria eating organisms are those viruses that infect the bacteria. Now examples of this, like some of the restriction enzyme. So BAM-H1, E. coli, Hind 3, PST1, SAL1, SMA1. So these are the different names of restriction enzymes, out of which this is extremely important. E. coli 1. Okay, so we are going to discuss this E. coli in detail. But do you people have any doubts or any confusion up until now?
No, ma'am.
Okay.
No,
All
ma'am.
right. So now you people can note it down.
Ma'am, we noted this, ma'am. We have to start from three basic steps in genetically modifying.
Okay.
Done, ma'am.
All of you are done, students?
Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am.
All right, did you people have any doubts up until now? Okay, now further we're going to see here you can see all the names, right? All the names of all the different kinds of restriction enzymes. But do you people know that there is a proper scientific way of this naming? There is a way of this naming. So we'll talk about how can we name a restriction enzyme. So we take the example of EcoR1. See, the first letter in any of the name of the restriction enzyme would denote the genus. The second and the third letter will denote the species. The next letter will denote the strain. And then the Roman number will indicate the order in which the isolation took place. Now, you people know that E. coli stands for Escherichia coli. Escherichia coli. So this is Escherichia coli R1. This is the name of a restriction enzyme. So always remember, always the first letter is for the genus, the second and the third is for the species, the fourth is for the strain. Now the strain can be rough, it can be smooth, and there can be a variety of more strains. And then the Roman numeral represents the order of isolation. In which order was it isolated? Now the working of restriction enzymes. Now, each restriction enzyme, it recognizes a specific palindromic sequence in DNA. Meaning, see, it doesn't mean that the restriction enzyme just goes anywhere and starts cutting. No. First of all, what it does is that it recognizes a particular sequence. And what is that sequence called? Palindromic sequence. Now, what is a palindrome? See, a palindrome nucleotide sequence, it is a sequence that would read, for example, if you're going from right side to left side and then from left side to right side, it would read the same in both the direction. For example, if you look at this one, 5 prime to 3 prime is GAATTC. And then 3 prime to 5 prime, if you read it from there, so it will be GAATTC. So from left to right and right to left, when it is read in the same way, we call it as a palindrome. Just like how I've here mentioned some English words to tell you what palindrome is. Mom, M-O-M. If you read it from here, or if you read it from the backwards, it's the same. Then dad also, D-A-D, right? If you read it from the front, like it is the same, and back also it is the same. Then Malayalam, see, it has the same spelling when you go from left to right, also the same spelling when you come from right to left. So this is what a palindrome is. So what the restriction enzyme does is that it particularly inspects the length of DNA. It sees what is the palindromic sequence and then it starts cutting. Also remember one thing, that each of the restriction enzyme, they inspect the length of DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and then it will cut each of the 2 strands of the double helix. Meaning, it will first of all inspect the length. Once it will find its sequence wherever it is supposed to join, then it will bind to that DNA and then it will start cutting the strands of the double helix DNA. Now, there are 2 types of cuts that are made by the restriction enzyme. So just like how if you cut paper, if you cut it very nicely and very precisely, you'll have a straight papers that are there. Then if you cut it in a haphazard way, then you will have a very useless kind of a paper cut. So here, the types of cut that are made by the restriction enzymes are sticky end and blunt end. Now, when the restriction ends on endonuclease or enzymes, they cut the DNA a little away from the center. When the cutting is not done in the center, rather a little away or shifts towards like away from the center, then sticky ends are generated. And these sticky ends, you can also call them as staggered ends. But whenever a restriction enzyme is cutting the DNA right in the middle of the palindrome, so it will generate a blunt end. Also, always remember that the vector, which is the carrier, right? Carrier of like the gene of interest and the source DNA, whatever we want to put on the vector to be carried, they both have to be cut by the same restriction enzyme. If it is not done, then both of them will never be able to form a bond. So the vector and the source, they have to be cut with the same restriction enzyme. Then only the recombinant DNA will be created. Now, we know that we have cut the gene of interest and we have cut the vector. Now we want to join them together. So we can't just join them like that. We have to use, because we were playing with the scissors, so now we need the glue. So DNA ligase, the second enzyme, right? It is a molecular glue. What does it do? It joins the fragments of DNA by forming a phosphodiester bond. Now this again reforms the phosphodiester bonds. Any doubts?
No,
Okay, now you people can note it down.
ma'am, I have a doubt.
Yes, tell me.
In the naming of RE, for
Hmm.
example, BAMHI, the H letter, what type of strain is that, rough or smooth?
The strain, the edge. See,
Yes,
the edge can see, as I told you that the strains can be of multiple types. See, we only know the rough and the
smooth.
smooth type, right? Now, the edge strain, it is basically referring to a strain that is sort of like antibiotic resistant strain, meaning a strain, or you can say a strain of bacteria that is resistant to variety of antibiotics, meaning it will not, you can say, undergo the antibiotic effect. That is what the H strain represents. Okay.
Okay, ma'am. Thank you.
Same way, like it will happen with all of them. Some will have the strain mentioned with them and some will not.
Thank you, ma'am.
Okay.
Ma'am, can you go down? Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am.
Done.
Done. Yes, all of you are done.
Done, ma'am. Yes, ma'am. Done. Done.
All right, any doubts?
No, ma'am.
All of the things taught today are clear to you all?
Yes, ma'am.
All right, okay. All right, so today we have learned the various enzymes that are involved in the process of recombinant DNA technology. And now further on, we'll see what the vectors are, how the vectors are transferred to the host and everything about that. Now, I'll keep it till here only as far as this chapter goes. Now, you people tell me, which chapter are you currently studying or learning for your test or anything of that sort?
Human reproduction, ma'am.
Human reproduction. Okay, what about the others?
Ma'am, I'm also doing human reproduction.
Okay, so all of you are doing human reproduction chapter. All right. So let's just do a quick revision of human reproduction. Students, tell me one thing. I'll ask you people the questions from that chapter. Let's see how much you people retain or remember, because we have done this chapter long back, isn't it? All right. Now, first, the easiest question. What are the male and female gametes in human beings?
Male gamete is the sperm, and in the female it's the egg.
Okay. Is the
Egg, ovum,
yes.
ovum slash egg.
Okay, what are the components of the female reproductive system?
Two ovaries, two fallopian tube, uterus, uh, cervix, vagina,
Yes,
external genitalia, mammary glands, two mammary glands.
very good. Exactly, very good. Okay. Now tell me, how many seminiferous tubules are present in the males?
250.
250.
Uh,
250 testicular lobules are there. But I'm talking about seminiferous tubules. If someone is saying anything, please speak a little louder because I can't hear you. Khadija, are you saying something? See, there are three highly coiled seminiferous tubules. Three of them. Okay, can someone tell me why is why are the testes located outside the body of the male?
because for the formation of
Um,
sperm, a lower temperature lower than the body temperature is required.
Okay, and what is the body temperature?
37 degrees Celsius.
Okay, very good. And those testes are present in a pouch-like structure. What is that structure called as?
Scrotum.
Okay. Okay, what is the function of vas deferens? The function of vas deferens.
provides Ca and phosphorus ions to the sperm.
Okay. If that is if the vas difference is doing all of that, then what will the associated glands do? Those things that you're saying, the secretions and everything that are released by the male glands. Then what is the function of vas difference? Basically, it is responsible for the transportation of the semen or the formed sperms. They are transported with them. Okay, what is the testicular hormone name? The
Testosterone.
testicular. Hmm?
Testosterone.
Hmm. Basically, the androgens, meaning the male hormones that are released. Okay, which hormone is the hormone that is released only during pregnancy in the female? Only during the pregnancy.
HEC,
H,
HCG,
HCG. What is the full form of HCG?
human cross,
Say. Chorionic.
human chorionic gonadotropin hormone.
Very good. Exactly. This is the hormone that is released only and only when the female is pregnant. Okay, tell me what are the three parts of sperm?
The head,
Middle, and tail.
middle, and tail.
Okay, there is also a neck, right? Neck is also there, a tiny neck. But I asked three parts, so you people are right. Okay. What is the difference between primary oocyte and secondary oocyte? Like, is there a difference between them?
The first primary oocyte is in meiosis one,
Hmm.
and
Correct.
the second primary oocyte is in meiosis two.
Secondary oocyte, yes.
The
Then
first one, the first one is surrounded by granulosa cell. No, sorry. Wrong, sorry.
before that, it is which one out of them is arrested at the prophase one?
Primary.
Okay. Okay. Tell me what is the meaning of fertilization? Fertilization, meaning of fertilization. All of the tough questions you people are able to answer, but the easiest one you don't know.
The fusion of male and female gametes.
Yeah, exactly. The fusion of the male and the female gamete is known as fertilization.
That that's the only definition.
Yes.
That's that will be enough.
Yeah, the
Oh,
fertilization refers to the process wherein the male gamete, that is a sperm, fuses with the female
fuses with the
gamete,
female
that is
gamete.
a
I thought I had to go in depth of it, so I was remembering all the notes.
no. See, when you are being asked about all of the like you can say pre-fertilization events, then you'd have to tell us. But right now, it just has fertilization. That has a simple answer. Okay. Tell me what is the meaning of parturition?
Sorry? Rejection of the fully formed delivery of the baby.
Yes. Both of you are correct. The delivery of the fetus is known as parturition, right? And parturition takes place because of a reflex. What is the name of that reflex?
Neuro.
The name of the reflex.
Oh, the reflex.
Hmm. Fetal ejection reflex, right? Fetal ejection reflex. Now, this reflex, that is fetal ejection reflex, it is like, where does this reflex come from? Who gives the reflex? Two of the components, they give the reflex. What are the two components?
Uterine muscle contraction.
No.
Ma'am, could you repeat the question?
The question is, the fetal reflex, the fetal ejection reflex is given by which of the two things? It is actually given by two components. So which two components?
Oxytocin.
Hmm?
Oxytocin.
No. The fetal ejection reflex is given by the fully developed fetus and the placenta. These two components, they give the fetal ejection reflex. Okay. Which hormone is released during the parturition?
Oxytocin.
Oxytocin.
And oxytocin causes what?
Muscle contraction.
Of?
Uterine wall.
Very good. Okay, what are the three walls of the uterus?
Endometrium, myometrium, and perimetrium.
Endometrium, myometrium, perimetrium.
Okay, which of these layers undergo the cyclic changes?
Endometrium.
Okay.
Endometrium.
Which is the part wherein the zygote gets implanted at?
Fallopian. No.
Hmm.
Uterine.
Endometrium.
See, if I say which part of the female reproductive tract, so you'd say uterus, because that is where
Oh,
the zygote implants. But where does the fertilization take place? So in the fallopian tube.
hello.
And at which part of the, like, there are three parts of fallopian tube. What are the three parts?
Ampulla, infundibulum,
Yes, and
ampulla, isthmus.
yes. Okay, what is that structure which you can say sort of catches the ova as soon as,
Finger-like
yeah.
membrane.
Okay, very good. Do you people remember the pathway of milk ejection?
Mammary ovules, then to mammary
Mammary ovules.
alveoli. Mammary
Okay, mammary
alveoli,
alveoli.
mammary duct,
What forms a duct? What forms a duct? Huh? Correct. So first, mammary tubules are there. Then many mammary tubules, they form the duct together. So like it becomes a mammary duct. After that,
mammary ampulla,
mammary ampulla. And last is,
lactiferous duct.
very good. Exactly. Okay, you people are preparing very nicely. I'm very happy. In the end, you people have made me very happy. Okay. Okay. We know that the placenta is present inside the body of the female. What is the function of placenta?
It transfers of nutrients to the embryo
Hmm.
and antibodies.
Also, tell me this thing. The placenta is connected to the fetus via?
Via uterine wall, uterus wall.
No.
The umbilical cord.
Umbilical cord.
Umbilical cord.
Okay. Okay. What is the difference between menarche and menopause?
Menarche is the start of menstruation
Menarche is the
and
first.
and the Menarche and last
Menopause.
monarch, right?
Menopause is the
Menopause
last
is
when it
stop
stops.
Yes,
at
it
the age of
stops
45 and
permanently,
50.
right?
Yes, ma'am.
Very good.
And
Exactly. Okay. All right. What is the difference between spermiogenesis, spermatogenesis, and spermiation?
Uh,
Take it slowly.
spermiogenesis,
Let's start with spermatogenesis. What is the meaning of spermatogenesis?
sperm,
for
Eh?
formation of sperm.
formation of sperm,
Hmm. Formation of spermatocyte is
spermatocytes.
spermatogenesis, right? Okay. What is the meaning of spermiation?
The release of sperms
The release of sperms from
from the testes.
Ejaculation duct,
no, not like in the testes only. Which part of the testes?
I um,
ma'am, what's the word again? Sperm?
Hmm?
What's the word again? Spermiation.
Spermiation. Spermiation. It is the release of the you can say the formed sperms from the seminiferous tube. Okay. And the last one is spermiogenesis. What is that?
Spermiogenesis.
Hmm.
The com the complete process of sperm formation.
That is spermatogenesis.
Oh,
Spermiogenesis is the transformation. People remember they were
yes.
getting embedded into the heads of the
Yeah.
Sertoli cells. They were getting transformed. Right? That is spermiogenesis, wherein the spermatids were getting transformed to spermatozoa. See, spermatozoa or sperms, it's the same thing. Okay, tell me. Okay, last question. There are two gonadotrophins in the body of males and females both. What are the gonadotrophins? Gonadotrophins. I'll give you one hint. They both are hormones. And they are collectively called as gonadotrophins. So what are they? Tell me the names.
Estrogen and no,
No. There are only two gonadotrophins. Both of them are common in male and female.
LH and FSH.
Correct. LH and
Luteinizing hormone and follicle stimulating hormone.
stimulating hormone.
very good. Correct. Okay, so your preparation is good. All of your preparation is good. Nice. Very nice. You people keep on reading the NCRT, the notes as much as you can. And you people have retained a lot. A lot of the information has been there in your brain. It's very good. So you people keep on practicing more and more. You'll have a very flawless memory of the whole entire chapter. I'm very happy with your performance. So just like that, slowly, slowly do all of the chapters or according to your syllabus, whatever is coming, do all of those. Okay. So once, like, what is the next chapter that all of you will be tackling after human reproduction?
Uh, Human health, ma'am.
Reproductive health.
Reproductive health, yeah. That's
Reproductive health.
Yes.
All of you?
Yes.
Yes.
Okay, so tomorrow we have another class, right? So at the ending of tomorrow's class only, I'll ask some questions from reproductive health just to help you. It's not a test. I'm not judging you. I'll just see how much you remember if you have any